So from this example, we now see that the dynamic changes of connectome can explain the emergent functions, and also some of the macroscopic functions may emerge from the coordination across the multiple microscopic phenomena. So we need to understand the coordination process. Lastly, there may be some common principles. So it's maybe important to understand all these phenomena in a quantitative way, and we eventually want to understand the underlying mathematical principle that controls this kind of phenomena. So what we want to do here is to record the changes of the connectome quantitatively. Next, we want to see the functional changes accompanying the structural changes. And lastly, based on the quantitative data, we want to understand the underlying principles. That is our basic strategy. So for this purpose, we would like to use some of the cutting-edge technologies we have. For example, this is electron microscopy-based connectomics, and this will be supported by photosynthesis group. This is a ultra high-speed 3D EM that is quite efficient in taking a large volume of images using EM. Also, that photosynthesis is very good at combining the photon in vivo imaging and EM structured imaging. So that can be quite powerful strategy to correlate the static EM images to the dynamic photon images. And second thing is light microscopy-based structured imaging. Our laboratory has been developing tissue clearing and labeling strategies, and that can be useful for looking at fine-scale connectivity even with EM. Light microscopy has some good advantage. For example, the compatibility with the genetic labeling. So we think that this can be a very powerful strategy. Also, our laboratory is supporting the light microscopy-based whole brain imaging. And this is an example of the multiplex imaging of the whole brain. And we also support the quantitative imaging of the whole brain like this. So this can be an important platform to facilitate the whole brain scale dynamic connectomes. Also, the Maruyama-san's group has recently developed the large field of view to photomicroscopic objectives like this, very cover a very large area of the cortex. This can be quite powerful for the acquiring comprehensive dynamics of the cerebral cortex. So we'd like to support all these kinds of techniques to facilitate the collaboration within our groups. And here is all the group members. In AZ1, these 4 PIs work on the emergent function in the neurons. And in the next category, Maruyama-san, Kota-sensei, Sato-san, Miyazaki-san will work on the circuit scale of emergent functions. And lastly, the 2 PIs work on the reconstitution or the computer simulation of this kind of behavior. Also, we support technology parts. And also, lastly, importantly, there are 4 advisory members, including Mara and Yamamoto Sensei. And we really appreciate your feedback. Lastly, this is important for maybe people from outside our groups. And actually, the public call for the proposal is now opening, and the deadline is maybe in September. And there will be 21 slots for the public call. So please, I'd like to encourage everyone to apply for this slot, and we really look forward to the collaboration. And we like to support original and also the synergistic and complementary project to our ongoing projects. But our group currently cover very limited area of the dynamic connectome. So we really appreciate the complementary ideas, including the functional changes during the life cycle. Because many of us work on the developmental steps or the learning process, but we do think that the other kinds of dynamic connectome is important, such as the changes during the life events, pregnancy, or the adaptation to the environment, or aging, and so on. Also, we do think that the nervous system is not limited to the brain. It's connected to the every part of the body, and that is quite important. So, yeah, systemic regulation is another important target. Also, we do respect the study in other model organisms, like from Drosophila to the primates. Also, we think that the some problem in a dynamic connectome may have important connection to the diseases. So we do support a proposal related to the disease. Also, in the future, maybe the neuroscience and AI will be merged in the future, and we think that it's important to understand the emergent phenomena happening in circuit exchanges and reconstitute that in a mathematical way in the future. Okay, so that's all from me for now. And thank you for listening. And I'd like to move to the scientific part. Thank you.